Dr. Akanksha Mishra. Basically, she was supposed to be here yesterday because she is the recipient of Milan Pharmaceutical Brightest Young Indian Scientist Award. So she was late yesterday, so she will be presenting a paper today on purification of recombinant human PLC zeta as a therapeutic modality for oocyte activation deficiencies. Giving a brief introduction about Akanksha Mishra. She is an IVF laboratory director at the Sabine Hospital and Research Center, Kerala. She has done her MS in clinical embryology from the University of Oxford, UK, and has experience of over 10 years in the field. She is on the International Advisory Editorial Board of IVF Light Journal of Minimal Stimulation IVF. She has several journal publications and chapters contributed to her credit. She has been an invited faculty at several workshops and conferences. Ms. Akanksha Mishra, please.
Let me take this opportunity to thank CRT organizers, Dr. Alabadia, Madam Gurul Gandhi, and everybody else for giving me this opportunity, and Milan for considering me for the award. As mentioned, I was supposed to present this yesterday, but unfortunately I could not. So the topic is purification of recombinant human PLC zeta protein as a potential therapeutic for oocyte activation deficiencies. This was the topic of my research work while I was pursuing my MSc. This still happens to be a continuing research work. It wasn't conclusive. In fact, this is a research work that has been going on for the past decade. About 3 major groups and a lot of minor ones are trying to isolate the PLC zeta protein. So let me begin by apologizing to you that I will not be able to share the exact method that was employed in purification of the protein because I don't have the permission to do so. Now, I don't know why this protein has remained so elusive for the past decade, but it is very important to know the significance of the small 70 kilodalton protein and why exactly these groups are working so hard and so much and for so long to isolate it. So before I begin, let me draw your attention to this slide about global fertility statistics, because this is very important. Just keep the slide in mind. The battery is low, please. Now, the factors causing fertility, a lot of it is contributed by male factor. ICSI has proven to be a way to overcome the male factor infertility issues. But even while we inject one sperm into an egg, we notice some failed fertilizations. About 25% of the cases, we see failed fertilization. Now, out of this failed fertilization, most of it is partial failed fertilization, but about 10% of the times, you also see total failed fertilization. So why is this when you're actually injecting a sperm inside an egg and there is a total failed fertilization? Let me take you back to the basics. The main events of oocyte fertilization. You have the capacitation, you have the acrosome reaction, the membrane binding, the sperm nucleus is injected into the egg, and the egg nucleus and the sperm nucleus, they fuse together. But there is something that happens here, something that happens. Some signaling has to be given to the egg nucleus so that it knows that the sperm has entered and it has to start the process of fusion, which is basically called the egg activation. Egg activation is when there is release of cortical granules, when the egg is released of the M2 stage, and the pronuclear formation occurs. Throughout the years, various scientists have tried to find out which exactly was the molecule that was, or the element that was causing this oocyte activation that was playing a major role. So all these various scientists, starting back from 1937, working on different species of eggs, found out that the calcium plays a very important role in the oocyte activation. As you can see here, these are calcium oscillation studies. What these scientists working with different species of eggs observed was that the moment a sperm enters the egg, there is some sort of calcium increase or a pulse or a wave of calcium that occurs inside the egg, which causes the activation to occur. This is a predecessor for activation of the egg. As you can see in different species, the waveform is different. It varies from species to species, but it is present nonetheless. A number of hypotheses have been conducted, have been proposed over the years to find out where exactly this calcium comes from. What is the source, whether it is coming from outside, whether this is a sperm calcium, or whether it is the egg calcium. So first was the sperm conduit hypothesis proposed by Jaff. According to this, what they said was that the sperm, while it undergoes activation and acrosome reaction, the membrane destabilizes. It takes the calcium from the medium and it acts like a conduit. When it enters into the egg, it carries the calcium along with itself. But this theory was subsequently disproved because the activation was, egg activation was found to occur even when the egg was fertilized in a calcium-free medium. So the sperm could not have been a conduit. Second was a membrane ligand model. According to this, they said that this receptor, egg receptor on the sperm, have a protein called as bindin. And this is the protein that sends tyrosine phosphorylation signal, and this causes the egg activation. But the major flaw this theory had was that it depended on the membrane interaction between the sperm and the egg. Whereas it was also obviously observed that when you're doing ICSI, you see fertilization. So there is no membrane interaction over there. So this theory was disproved. The final theory was sperm content hypothesis proposed by Swan in 1993. It says that there is some proteinaceous wave-inducing component of the sperm cytosol that diffuses into the egg while fertilization, and it causes the release of the intracellular oocyte calcium, which induces the calcium oscillation wave. Now that the soluble sperm factor theory was firmly established, there were several candidates that were proposed to be that particular sperm factor. It was truncated form of C-tyrosine kinase receptor, but this was mostly found to be in the mid-piece of the sperm rather than the head, so this was disqualified. Next was citrate synthase, but the actual mechanism of action of this molecule was not known, not understood. This was post-actosomal sheath WW domain binding protein, or PAWP. This was a more likely candidate, and even till date it is it is believed to work parallelly along with the zeta in oocyte activation. Now the molecule that we are interested in, PLC, phospholipase C, it was discovered by Parrington in 2001, way back in 2001. He did not discover the exact isoform that was responsible, but just the phospholipase C. Just to give you a brief idea, this is the structure of human PLC zeta protein. But this can get a bit complicated to understand. So I have created a schematic diagram of this. It has the EF hands, which are in green, the red, which are the catalytic domain, X and Y domain, and this is the C2 domain. They all have their individual functions, but the most important are the catalytic domain. Now, I will not go into the exact detail of how every domain functions, because that's not really important or relevant right now. But I would like to mention that zeta is the smallest isoform of all PLC proteins. The mechanism of action of PLC zeta was described very well. Upon sperm-oocyte membrane fusion, the PLC zeta diffuses inside the egg cytosol. It hydrolyzes the membrane-bound phosphoinositidal bisphosphate to DAG and IP3, inositol triphosphate. PKC is observed, and IP3, it acts on the endoplasmic reticulum, which is a calcium source inside the egg. the endoplasmic reticulum channel, calcium channel open, the calcium is released, and egg activation and cell cycles resume. Now, since PLC zeta is not available still, for oocyte activation deficiencies, some artificial oocyte activators have been used already in several labs across the world. There have been calcium ionophores, isoleucine, puromycin, ethanol, strontium chloride, phosphol esters, and even electrical stimulation. They do cause some degree of calcium oscillation inside the oocytes, but they do not mimic the natural calcium oscillations. They are not physiological, they are artificial, and there are questions as to their biological safety and epigenetics. Now, several scientists have observed that the injection of PLC zeta into an oocyte causes calcium oscillation, which is similar to natural. There have been several studies that have been conducted to emphasize this theory that PLC zeta is the one that is actually responsible for oocyte activation. The sperm which are devoid of PLC zeta, they fail to activate oocytes. And this oocyte activation failure was rescued by injection of PLC zeta into mRNA. PLC zeta mRNA into the oocyte. If there are any mutations in the PLC zeta, it also abolishes the calcium oscillatory ability of oocytes. And the PLC zeta, this paper mentions, if you would like to check it out, the relationship between oocyte activation and male factor infertility. So by now, it has been firmly established that any mutation in PLC zeta causes loss of activation. So this is definitely an oocyte activation factor. So the aim of my experiment was to express and purify recombinant human PLC zeta by various methods and confirm its presence by electrophoresis followed by Kumasi blue stain, silver staining, and Western blot, as well as by LCMS studies. Briefly, materials and methods for protein purification. The human embryonic kidney cells were transfected with wild-type PLC zeta open reading frame. There has been a group, Yoon et al., a Korean group. They have already tried transforming bacterial cells and tried to produce PLC zeta, tried to get the E. coli cells to produce PLC zeta. But they were not that effective. Probably the reason is that in bacterial cell, the folding, the final folding of the protein does not happen. The maturation of the protein does not happen. It is just released as it is. So maybe if we use a mammalian cell, we will get the protein in its native form. This was cultured for 4 days, isolated and purified the protein. It was studied by staining and calcium oscillation studies. These are the results that I got. As I mentioned, it's a 70 kilodalton protein. So we did observe the bands in the silver staining and the Western blot method. But as you can see that this is not very pure in the elute that we found. You can see several bands along with it. So this was not a very pure protein that we got in the end, which was our objective. So although I could not, because of the limitations of time, pursue this further, but my research paved its way for future work, which includes purified protein to be subjected to LCMS, mass spectrophotometry, and calcium oscillation studies to find out the purity of the protein. Injection of the purified protein with the isolated sperm nuclei to detect fertilization and embryogenesis. And third is modification of culture purification conditions for more efficient protein production. I would like to end by acknowledging my entire team and group at Oxford with whom I conducted this research. And thank you all for your patient listening. Thank you.